very excited to introduce Claire Hales, who is a postdoc in the Winstanley Lab, who's going to be talking about computational modeling of gambling in rats, bridging the gap between rodents and humans. And Claire, I'm going to bore you by bore the group by telling a story I always tell, which was when I was sitting on a dissertation defense, it must have been one of CAF's students. And Chris Moll was a university examiner. And so the student presented on the rat gambling task and things that had been learned on that. And we went around for Christmas and Chris said, "Do rats gamble in the wild?"
I mean, I think probably gamble in a sense that they're gambling with their life, right? Like they have to make decisions.
Oh, good answer.
About, like, where to go and forage. Like, am I going to get like run over by a car? Like, but I don't think, yeah, I mean, they definitely don't gamble in the same way as humans do with money.
But that's a great answer. That would have been a great answer for the defense. So I'm going to give it over to you, Claire. I have to step out, but I'm very happy you're recording the talk because I will then be able to listen to it later. So Claire, you will have the power to stop recording when you think it's appropriate, when the whole event starts to disintegrate. And everybody, please do remember to eat all the delicious food that I believe is sitting in the corner. And yeah, looking forward to seeing everybody back in September.
Great, thanks very much, Beck. I am going to share my screen, hopefully get that to work. So can everyone on, no.
Yeah, I see the presenter view.
Yeah.
Right, we see, yeah, not the...
Like switching it over, isn't it? And there's a way to do it, and I always forget how you...
Yeah, I always forget how to. But Brandon probably has some suggestions.
Well, what I would recommend basically to just triage this, do you have something up on the screen behind you? Like, so are you connected to, like, basically to the TV, right? And
yeah.
So, and is it set up currently so that you have like on your TV the slides that you want to show?
Yeah.
Okay, so the solution would be to unshare your screen and go back to the screen sharing options and select, it should be desktop 2, the one that has your slides in it, not like just your window of PowerPoint, but desktop 2. And then start presenting again. And you might have to flip back and forth between presenter view and the normal view, but that should work. So check if there's like desktop 2 that's listed there. I'm not sure if you have like a mirror to display or if usually there's a second to display, but if you have something like desktop 2 which shows your slides, that should be it.
Or maybe screen 2 or something like that.
That's what I did last time, that just shared it as it.
Okay, so I guess if you go into the presenter view now, like if you start presenting it from the beginning, yeah.
Ta-da! Yeah, anyone who's on Zoom can see.
Okay, so now all you have to do is click like swap the displays in this window here. We can see your presenter view, so just go to display settings in like just at the top of the screen here, or the option, whatever the options for swap displays. I think display settings is where it is. So no,
yeah.
it's at the top of the screen.
Yeah, display settings, like a drop down.
Display settings, swap. Yeah, that one. Yeah. Perfect. Now we can see.
Now we can see presenter in the room though. I swear this happened last time, right? Did we remember? Did we?
You're okay. You're probably sharing the wrong screen. Then you're probably like sharing the screen that is on your desktop, like on your own computer. Then, so you need to go back and make sure that you are sharing the screen that is actually the TV screen, as opposed to your computer screen. If it appears, it should appear normally. Like there should be, it should say like desktop 1, PowerPoint, something, and then desktop 2.
Like it's not, it's like I've only got screen and nothing else. I mean, I could just do it with this, but because that's the other way. No, because it's the other way around. I mean, I could just make it like kind of full screen, but not actually full screen. How do I unpin? What about if I just do it like that?
It looks fantastic to me, but yeah, it's up to you.
Do it like this. Yeah, I feel like we might spend a long time trying to figure it out.
Yeah.
Awesome. So okay, great. So I am going to talk to you about the way that I am attempting to help to bridge the gap between rodent work and human work in gambling disorder using a couple of, well, one main approach, computational modeling, but combined with some translational tasks. So I need to now just click to, there we go. So just to set the scene, gambling disorder is a psychiatric disorder that a proportion of people develop. But the really interesting thing, or one of the really interesting things is, lots of people participate in gambling recreationally, but not all of these people end up developing problems that are significantly impacting on their kind of life or health in a negative way. So, 75% of all Canadians participate in some form of gambling, but for the majority of those, there's absolutely no problems. However, for a small percentage, this does develop into having severe consequences on their, personal, professional, social lives, and as well as people that meet the actual DSM criteria for gambling disorder. There's another percentage of the population that we would classify as, showing very risky gambling traits that could well be, are those that are likely to or could easily develop into gambling disorder. And just as this fact kind of blew my mind, was like, the most serious gambling addicts often lose more than $500,000 a year, which is a crazy amount of money. And kind of makes sense as to why this has this negative consequences to people. So we know from lots of research that there's lots of cognitive processes that have been associated and linked with problematic gambling. People have changes in their sort of cost-benefit decision-making processes. There are reward and risk processing changes, particularly to do with loss aversion. We know that impulsivity and compulsivity are linked to gambling disorder, and there are changes in cognitive control and cognitive flexibility. And a lot of this research, at least that's done in humans, we often measure these things using behavioral tasks and or questionnaires. So people will come into the lab, they will play either gambling type gaming machines, or do decision-making tasks or other cognitive tasks that will measure these various kind of attributes. And then we can also combine with that, use questionnaires to ask them about their sort of traits, states, what they're thinking, what they're feeling, why they're perceiving things in a particular way. And this is great, but if we want to kind of dive down into the neurobiology and try to understand more about what is happening kind of in the brain in terms of circuits, neuroscience, neurotransmitters, then often we want to use animal models. And so animal models in psychiatric research, like there is a lot of them, but one of the big kind of issues with animal research is how beneficial or not they actually are in psychiatric research. So some of the main benefits are that we can directly manipulate the sort of neurobiology, the brain circuits and the neurotransmitters, and as well as direct manipulation, we can also directly record from brain activity whilst animals are performing the sorts of tasks that we're interested in, or exhibiting the sorts of behaviors. There are feasible, like other constraints we have to think about with animal models, feasibility, like practical kind of measures, economic factors. But in a lot of cases, we can ask questions in animal models that we just can't with humans. However, there's also some quite big limitations. We can't just ask an animal why it's doing what it's doing, or why it's feeling or thinking what it is. We have to measure their behavior and then interpret what that behavior means in relation back to the sort of thing of interest that we think it relates to in humans. And we all know that across psychiatric disorders, translation from animal research to the clinic can be tricky. There's lots of, particularly in for pharmaceuticals, drugs that have shown efficacy in animal models, but then haven't gone through to have that same effect in humans. And so I don't know if anyone saw Djavad Young's talk a couple of weeks ago at the Neuroscience Research Colloquium, but I basically stole this directly from him because I really like the way he was talking about it. You know, this sort of the depth valleys in, you know, translating kind of what we do in sort of basic biomedical research with animal models through to things that are useful, both just in terms of knowledge and how we can apply that to humans, and then the next step of then making that knowledge relevant and, you know, able to be impactful in a clinical practice and or for health outcomes. And so what I'm going to hopefully sort of convince you of today is that one of the approach that I'm taking is one way that we can try and bridge this gap between on value one, essentially. So, you know, using translational tasks and computational modeling to try and make it easier to take the results and what we're learning from animal models and apply this to humans. So I'm going to focus on a couple of translational tasks that were developed here in the Winstanley lab. So the rodent gambling task, which is a version of the human Iowa gambling task that measures risky decision making. I'm also going to talk about the rodent cognitive effort task. This is a task that, unlike a lot of previous tasks in rodents that actually use physical effort as a measure of kind of effort-based decision making, uses a cognitive measure instead. And then I'm going to tell you about sort of the work that I'm doing here, which is using computational modeling to try and get, you know, more understanding and more translational relevance from the animal tasks that we're doing and the manipulations we're doing in animals. So here in the lab, we use two main approaches, reinforcement learning modeling, but I'm not going to talk about today because that's other grad students that are kind of have been pioneering that work. But what I am going to tell you about is the use of a cognitive model, the drift diffusion model, which is a two-choice sequential sampling model that I am using to essentially try and give us more insight into what it is that rats are doing when they are performing these tasks. So before I go on to tell you about more detail the tasks and the model, I first of all want to motivate why I think this is a good approach with an example from my PhD work back at the University of Bristol. So I developed a reward-based decision-making task that essentially enables us to ask a rat the kind of prototypical question of, if you have a glass that is filled halfway, do you interpret that positively, as in that the glass is half full, and do you interpret it negatively, as in the glass is half empty? And the reason that this is, you know, potentially useful to ask a rat is that this kind of ambiguous cue interpretation is affected by, or is changed by affective state. So we know that people that are in positive moods are more likely to be optimistic, and people in negative moods are more likely to be pessimistic. And this kind of affective bias is really relevant in anxiety and depression. And so the task that I developed essentially was a reward-based ambiguous cue interpretation task, or judgment bias task, where we trained a rat that a particular tone meant that if they pressed the corresponding lever, they got a high value reward, whereas a second tone, if they pressed the other lever, they got a low value. So one of these options is more positive, one of these options is less positive. And then we essentially asked the rat, glass half full or glass half empty, by presenting an ambiguous midpoint tone and getting them to press a lever. We can count how many times they press the high reward lever versus the low reward lever, and that gives us a measure of sort of, yeah, their optimism and pessimism. So the one thing that I'm going to tell you about here is a manipulation we did to induce anxiety, so to induce a negative affective state. In this case, we used injection of an anxiogenic drug, FG7142. But the kind of critical bit here is that, you know, not that we just did this in rats, but that we also did the same thing in humans. So I collaborated with a lab at UCL to essentially, in this case, reverse translate the task that I developed in the rat back into humans, mainly because there are lots of examples of tasks that measure ambiguous cue responding in humans, but pretty much all of them use either ambiguous faces, so, you know, smiley faces, through to unsmiley faces, unhappy faces, which obviously we can't do with a rat, because a rat can't read a human's facial expression, or at least we think they probably got. Or they use ambiguous words, but again, we can't do language with rats. So yeah, we translated this task to basically exactly the same format, but just humans were getting, instead of large and small food rewards, they were getting large and small money rewards. And instead of injecting them with an anxiogenic drug, because, you know, ethics not necessarily able to do that, we used a different approach where we interviewed them using the MINI, so the MINI International Neuropsychiatry Interview, and counted up essentially, you know, how many affective related responses they had, and designated people as either being asymptomatic, so showing very little anxiety or depression related kind of problems or symptomatic. So this would be the idea being that they would have more negative effect and should show a more pessimistic bias.
Could I ask a quick question if possible?
Yeah, of
Just
course.
about the methods, because I'm having to do something similar. In terms of the tone for the large and the small reward, how did you, like, maybe there could be a bias in terms of their perception of the tone in terms of what's closer in perceptual space, so to speak, between, you know, the large reward tone and then the small reward tone. I guess I'm wondering, how were you able to figure out like a midpoint or a switch point? And yeah.
Yep, great question. So a couple of things. First, people back in the 60s and 70s did lots of work where they actually just tested rats' frequency alignments and got these like audio traces that show like how sensitive a rat is to a particular frequency, a particular decibel. And turns out, quite luckily for people that, you know, to do this sort of task, there is a proportion of their hearing alignment that is actually linear. So roughly between about 1 and 9 kilohertz. As the frequency increases, they get like less sensitive, or sorry, more sensitive, because rats are better at high frequency noises, which meant that we could essentially, you know, theoretically the midpoint should be a midpoint based on that research. But the other thing that we did, the way that we train the task, so we actually train it first that they learn tone A and tone B. They're both associated with one sugar pellet. And so what we can then do is play that ambiguous midpoint tone. And at this point, there's no reason that the rat should respond to anything apart from 50%, because there's no bias. You know, the reward isn't providing a bias. And so if they did respond to it not at 50%, that would suggest that there was a bias towards one of the tones just based on auditory properties. So we have that control in there as well.
Awesome. Thank you so much.
No problem. So what we were able to show is that, you know, both of these manipulations, despite being quite different ways of measuring or inducing anxiety between the rats and humans, caused this similar reduction in positive responding. So, you know, more pessimistic responding to the ambiguous cue. But not only that, when I applied the model that I'm about to tell you about to these behavioral data, the same model parameter, in this case, the drift rate, was explaining this change in bias. And so what this kind of, you know, it's quite nice when you get a translational behavioral result anyway between, you know, if you're using two different species. But what this kind of really made us confident was that, yeah, we are actually probably measuring the same process in rats and humans, because, you know, there's a lot of, you know, and quite rightly, people could argue, well, you know, do rats get depressed? Do rats get anxiety? But the point is that, you know, we're relative, even though, you know, rats probably don't get depressed in the same way as humans, we're measuring here something that is relevant in both rats and in humans. And so I'll go on now to explain the model and what this drift rate parameter actually means. So the drift diffusion model is a model that's been used extensively in human cognitive psychology literature. It was originally developed to explain perceptual two-choice decision-making, but in the last decade has been validated for value-based decisions as well. So the theory behind the model is that a decision between two choices starts at a particular starting point, and this decision and evidence is accumulated from this decision starting point to or towards one or other of the two choices, which are represented by these two boundaries here up at the upper and lower boundary. The rate of this information accumulation corresponds to how strongly the evidence for that decision is. So a steeper drift rate means that there is a stronger, you know, stronger evidence the decision is more easy for one option over the other. As I said, when the evidence gets to one of these two boundaries, that's the point that the decision is made for that choice. And the model also takes into account a measure for this boundary separation, which corresponds to essentially response caution. So, for example, if these boundaries are really close together, not very much evidence would need to be accumulated for one boundary to be hit, and it's much more likely that because this is a noisy information accumulation process, you could accidentally end up at the wrong boundary, as it were, or the boundary that isn't the decision that maybe would be the best kind of choice to make in that situation. The decision starting point corresponds to a sort of a priori bias for one option over the other. So if it, in this case, as is shown on this picture, you know, the decision starting point isn't neutral between the two boundaries, it's closer to one, suggesting that, you know, for whatever reason, there is a preference that that animal or human has for that decision choice that's represented by this top boundary. But the thing that gives a lot of strength to the diffusion model is that it incorporates both choices and response times when fitting these parameters. And so within the whole response time that you measure when you are doing a behavioral task, the model breaks this down into the actual decision response time, which is the time that this evidence accumulation process takes, but also this non-decision response time, which incorporates other factors such as sensory processing, motor processing, and other extraneous things that might be contributing to the response time that are not directly specifically the decision accumulation. And so essentially the way that the model fits is you calculate these response time distributions for the two options, and there's an equation with these parameters in that essentially recreates as closely as possible the shape of these response time distributions with different values of these parameters ending up, that would fit for making different shapes of the response times. So here you can kind of see like how it works in progress. So on each trial, this is an evidence accumulation path for each trial that you can see, and each one of these little ticks is a choice as the task is progressing and a person or animal is doing lots of trials. You accumulate all these kind of choices. And so essentially what the model does is work back from these choices to find out what would be the starting point, the rate of the average rate of information accumulation, the average boundary separation, and non-decision time to make these particular response time distributions. The particular software I use to fit the model can also fit some other parameters, mainly variability in some of these main parameters, and another bias term, but I'm not going to focus on any of these. They're not, I do validate whether they're useful in terms of fitting the model, but turns out that pretty much none of them ever are. So, oops. Okay.
So sorry,
go on.
can I just clarify, because I'm kind of a little bit confused on it. So what is the y-axis in this case? So is it like, because I can see it's kind of like a bumpy kind of mountain. Is that like, are you measuring something there? Is it like behaviors, or just like, are you taking a video? Is it their EEG, or I guess this is in rodents, you said?
Yes. So this isn't actually anything that I measure. This is just a schematic, like a schematic representation of how the theory behind the model. Okay. But saying that, it has been shown that, for example, in monkeys, if you record from the lateral limb to parietal area when they're doing a perceptual motion to motion dot task. So you've got lots of dots on the screen, some are moving in one direction, some are moving in the other direction. There are neural populations that show this ramping behavior. So it like this kind of noisy ramping like the drift rate is, which is, one of the reasons that it's kind of being used a lot is because we think that there are actually neural populations that implement this in the brain. But in this case, it is just like, I'm not actually recording any neurons to show that this is the case. It's an assumption of the model.
It's just decision time and then fitting the model to that decision time from the beginning.
Yep.
Okay, thank you.
That's okay. So I'm going to first share with you some, this is quite preliminary data from 2 versions of the rodent gambling task that we use, both the uncued and cued version. So I'll first of all take you through the task. So in the uncued rat gambling task, a rat starts a trial by nose poking into one of these kind of apertures, nose poke holes on the wall of the operant chamber. It then has to wait for a 5-second period before these 4 stimulus lights will come on in these 4 holes. If the rat makes a response in these holes before the stimulus lights come on, that we classify as a premature response, and we use that as a measure of impulsivity. But if the rat is able to wait and these 4 nose poke lights do turn on, each one is associated with a different probability and size of reward, and also probability and length of punishment timeout. So for example, for this hole, which we're going to call P1, if they poke their nose in this hole, they up for 90% of the trials, they will get a 1 pellet reward delivered. And then on the other 10% of the trials, they'll experience a 5-second timeout. Conversely, say on this hole, P4, they will get a 4 pellet reward delivered, but only 40% of the time, whilst on the other 60% of the time, they get a much longer 40-second timeout penalty. And so across the course of a fixed length session, a 30-minute session, the rat gets in the operant box to pick choices. The 2 options that I here circled in with the little yellow stars are the more optimal options, because even though they have lower pellet rewards, they're more frequent and the timeouts are shorter, and so they more often get to they'll more quickly be able to start a new trial and earn some more sugar pellets. Whereas these blue square options, P3 and P4, are the risky options, where even though they get higher per trial gains, they also are more high risk. because they're much more likely to get a longer punishment timeout. And so these correspond to the advantageous decks in the human IO gambling task, whereas these two would correspond to the two disadvantageous decks. And so that's the unqueued version. And then on the queued version, it's essentially exactly the same format. Contingencies are the same. The only difference is that on a win, the rat experiences a salient win-paired queue. And these win-paired queues scale with the size of the reward. So for the 1 pellet reward, it's a single kind of light and tone flash, like kind of boring, like not very exciting. Whereas for this big 4 pellet win, it's a whole series of lights and tone frequency flashing, much more like a electronic gaming machine or a slot machine, where, you know, a big jackpot and like all the bells and whistles go off. And so what the lab has previously shown many times over is that essentially animals that are trained on the unqueued gambling task show much more optimal profiles of decision making. So they much more often pick the two optimal choices, so P2 particularly and P1, versus on the queued task where animals switch to being more risky. So they less often pick P2 and more often pick P3 and or P4. And so Kath spoke last month at this mental health rounds and showed you some of the data from, you know, the latest really cool data from the lab, where we're looking at particular neurotransmitters, so dopamine, different brain areas that are kind of could be implicated in this, which I'm not going to focus on now. But one of the sort of kind of more basic questions that we have that is, you know, hard to answer is like, what is it about these cues that is actually driving this risky decision making? Like, why is it? What is it in the decision making process that is changing? And so that's one of the questions that I was hoping to answer using the computational modeling. And so just to quickly go through how I'm fitting the model, because if those of you that are sort of have been paying attention might be thinking, okay, well, you said that diffusion model is a 2-choice decision-making model, but rats here are making a decision between 4 choices. And this is a great observation. And so what I'm doing in this preliminary analysis, which is just to get an idea of whether, you know, using the diffusion model might be a good way to kind of find some interesting things that we can kind of look at in more detail, is to essentially recode either a P1 or P2 choice as an optimal choice and a P3 or P4 choice as a risky choice. So sort of making it essentially, on each trial, the rat either makes an optimal choice or a risky choice, even though they've chosen between 1 of 4 options. And so I'm putting in this trial by trial choice data and the response time for each of those P1 to P4 choices. And I fit the model to the final 3 acquisition sessions. So this is after the rats have learned these contingencies and are stably responding on the task. Because one of the things that the diffusion model is great for is to measure decision making, but it doesn't have any ability to, at least in its basic form, to measure learning about different options. So we're fitting it to after the rats have learned. And in this preliminary analysis, I've just fit to a single cohort in both males and females from both the unqueued and queued task. And the question that I'm trying to kind of see if I can get any insight into is, are there any decision-making differences between how rats are picking these optimal and risky choices, depending on the task? So are there differences in the queued versus the unqueued task? Are there differences depending on whether rats are naturally more likely to pick optimal choices or risky choices? And are there differences between males and females? And so in this case, I have selected that the upper boundary corresponds to a risky choice and the lower boundary corresponds to a optimal choice. So this is like all of the data together here, and this is a really complicated slide. So what I'm going to do is break it down and go through it one step at a time, so it's not quite so overwhelming. So what I'm showing you first is the parameter values for the drift rate. So this is the rate of evidence accumulation. And I've separated these out into males on the top, females on the bottom, and the different task types. So unqueued for the first 2 bars, queued for the second 2 bars, and then also split into whether the rats are optimal or risky. So whether they're whether on a session they are more often making P1 and P2 choices versus P3 and P4 choices. And so the first thing to point out, which is what we would hope for, is that optimal rats have negative drift rates down towards the optimal choice boundary, and risky rats have positive drift rates up towards the risky choice boundary. And if this hadn't pulled out in the analysis, something would be going wrong, because this is the same measure that I'm using to classify the optimal and risky rats. And so this is essentially just a sanity check to show that the model, like, it actually does work. But what is interesting here is that in females, there is no difference across task types. So optimal rats in the uncued task have similar drift rates to the cued task, same for risky rats. But in male rats, the uncued animals have much steeper drift rates down towards the optimal boundary than the cued animals. I will come back to what our interpretation of that in a moment. I'm now going to move on to the values for the decision starting point. So the dashed line on the graph represents a neutral starting point, which would mean that the rat has no prior bias towards one option or the other. And again, what we see here is a difference just across between optimal and risky rats, whereby in both males and females, taken, irrespective of task type, optimal rats have decision starting points that are lower than risky rats. However, what we can see is quite interesting difference between males and females. So here, up here, for the male rats, it's only the uncued optimal animals that have decision starting points that are actually significantly different from a neutral starting point, so biased towards this optimal boundary. Whereas in females, you can see that actually all of the groups, except for the uncued risky rats, have decision points that are biased towards the optimal boundary. And there's this particularly big difference in the uncued task, where the optimal animals have starting points that are very biased down towards the optimal boundary, and the risky rats have starting points biased towards the boundary. And so this is one of the things where I want to, add more animals into this, because I think that even though there's no actual differences here across males, it looks like there might be potentially this kind of differential where the cued rats in females are exactly the same, but maybe there could be a difference in the cued rats in males.
I mean, you probably get this all the time doing female stuff, obviously, but like, did you take into account the estrus cycle? And because I know there's some evidence in terms of like gambling and addiction, I think, and this is not my area of expertise, but where females during certain parts of their estrus cycle will be more likely to be risky potentially. So I'm wondering, is that something that you controlled for or looked into?
That's a really great question. No, it's not. We have, I believe, however, the lab has looked at whether estrus cycle, I mean, not in these rats, like the ones that I'm showing here, but we have before checked for that and not found any specific associations. Yeah, so we've found that estrus cycle does have an effect on impulsivity, but not on risky choice. So, I can't for definitely say that in this particular cohort of animals there isn't an effect, but generally we haven't seen an effect.
Thank you.
No problem. And so the other two parameters that that are the main parameters in the model, there were no difference between task types, so there are no differences between cued and uncued tasks. So I have condensed these here just for being able to fit it all on one graph and actually be able to see what the graphs mean. And we get another sex difference here, where in males the boundaries are the same between optimal and risky rats, but in females the boundaries are different. So optimal rats have wider boundaries than risky rats, suggesting that the risky females have reduced response caution. But across both sexes, we find a difference between optimal and risky rats in non-decision time, where risky rats have longer non-decision times than optimal rats. And so the interpretation of this, taking it all together, is that although we are measuring risky behavior, and from the behavioral measurements we take, it kind of looks similar between males and females, actually what might be driving risk preference in males and females across the different tasks is like a different kind of process. So to kind of try and like visualize this in a bit better way, I've created these kind of like, again, schematic representations of what these model parameters like mean in terms of the theory behind the model. And so what we can essentially see here is that the difference driving the sort of differences between the tasks in male rats is actually due to this drift rate, whereas uncued rats have these much more steep, optimal rats have much steeper evidence accumulation, so that it's easier essentially for them to make a decision for an optimal choice. And these optimal rats have much shorter non-decision times that compared to the risky rats. Whereas in the females, it's a different processes that is driving this. So the first thing, so these diagrammatic representations are to scale based on the parameter value. So you can quite clearly see the difference in decision boundary here, and how that even though sort of you're not getting a difference in drift rate, you can get a quite different sort of profile, because, for example, here with the positive starting point, the decisions are starting much closer to this risky boundary than they are, say, for the unqueued task.
So that's interesting. Sorry, I know I'm the only one who keeps talking. I don't know how many people are there, so I'm just you know, asking questions as I go along. So like,
Of course,
people in the room, you know, let me know if I'm talking too much. But it's really interesting, the idea of the decision time versus the non-decision time in terms of reaction time. And I think that on the previous slide, you showed that in males, the reaction time was different between two conditions, the queued and unqueued. Was that right on the decision time? Optimal versus risky, non-decision time. So
Yeah,
the
so
non-decision time was different, where it was took longer. So I'd almost think, like, I mean, this is just my intuitive sense, but like that, you know, risky rats would be taking a while to get decision, but then they'd almost be impulsive, and like, you know, their motor, you know, would be, you know, much quicker. But in this case, you found it the opposite. So I guess teasing apart the non-decision time versus decision time, how confident are you that, you know, that's actually capturing the non-decision time versus like the decision time could be kind of encapsulated in that?
That is a great question, and it's something that I want to go back to kind of check, as you say, to check how confident we are. So what I can say is that this is a result that, is counterintuitive, but has actually pulled out now across not only this version of the rodent gambling task, but has also pulled out in another version we do called the choice gambling task, where essentially within the task on each trial, the rat can choose whether they play a queued or unqueued trial. And so the fact that I found it twice across two different tasks in different cohorts of rats means that I'm, you know, that's one kind of suggestion that this is actually pulling out something that is real. But the way that we actually think that the reason that we think that it is counterintuitive, the explanation behind that is that although you might expect that sort of, for example, the motor processing part in an animal that is more impulsive / more risky might be shorter, you know, the non-decision time isn't just incorporating that, it's also incorporating, as I said, sensory processing, but other other factors that are just essentially kind of not the actual decision sort of time, so other irrelevant factors. And so actually what we think this might be is that what it's pulling out is that rats that are potentially more risky and or making more premature responses are more kind of distractible. They're not as focused on the task, which is what these longer non-decision times are kind of representing. But that is something that is at the moment sort of, I'm speculating, and one of the things that I want to sort of go back to do is to essentially kind of reverse engineer this modeling approach to say, okay, in rats that are the most premature rats, what do their parameters look like? Is it the case that premature rats, because at this point I'm not, you know, although it is the case that in the task risky rats can make more premature responses, it's not necessarily always that clear-cutter relationship. And so that's an analysis that I want to do in the future. So great point.
Awesome, thank you so much.
So essentially what is it showing is that, it's a different mechanism that, oh, sorry, go on. So the question was if I'm just looking at baseline data or if I'm looking at manipulation data as well. So this preliminary analysis is just baseline data. But what I want to do, which I'm literally about to put in a grant for that's going to go in by the end of the week, is to take essentially focus on dopamine. So say, okay, with all the chronic manipulations we've done with dopamine, where we've seen, during acquisition, we are able to change risk preference profiles differentially based on whether it's a queued or unqueued task, and also had some really interesting different results in males and females. Is potentially this could be a reason why we're getting those different results, is because the baseline behavior is being driven by a different process, and therefore dopamine, when we give the same dopamine manipulation, it's affecting males and females differently because it's a different process that is explaining the baseline. So exactly, that's what we want. yes, I think that was all I was going to say about this one. So moving on to the cognitive effort task. So again, just to take you through the task. On this task, this is a true two-choice task. So this is actually what I started off modeling, and the results here are from a much bigger cohort of animals, and also about to be submitted in a paper. But essentially on this task, again, the rat starts a trial by nose poking in the magazine tray. The levers extend. And in this case, the rat is making a choice between a hard or an easy trial. And so on hard trials, after the rat has made a choice for a hard trial, they have to wait again for this intertrial interval period. If they make a response during this time, we count that as a premature response. But if they're able to wait for that time, a stimulus light comes on in one of the 5 nose poke arrays in the operand box, but just in a single one of them. On a hard trial, this light comes on for a short time, so only 0.2 seconds, whereas on an easy trial, this light appears for a much longer time, so for a second. And the cognitively effortful part of this is that shorter time. So the rat has to make this visual spatial discrimination, which is potentially more difficult on the hard trials because it's a shorter, the light appears for a shorter time. If they're correctly able to detect this light, they get a reward delivered, which is greater on a hard trial than a easy trial. And if they don't kind of get the right nose poke, or they make a response, don't make a response at all, they get a timeout punishment. And so here, these are just circled to show you. So this is the data that's going into the model here, is purely the choices for which lever to press and the choice latency for that hard or easy trial choice. And so again, what the lab has found, which is similar to in humans, is that there is a proportion of animals that are willing to put in more effort and make more hard trial choices, which we call workers. And there are also animals that don't want to put in that effort and are happier, or will more often pick the easy reward choice, or less often than the workers at least. And the really critical thing here is that, within a single cohort, if we split these groups into workers and slackers, which was initially done based on a mean split, so we looked at the average kind of hard trial choice across the population, that was about 70%, and so we use a 70% cutoff for workers and slackers. We see that there is no difference in how they then perform on the visual spatial attentional part of the task. There's no difference in accuracy for detecting that stimulus light that comes on in one of the holes between workers on the hard choices and slackers on the hard choices, and vice versa for the low choices. And so here, as I said, I fit the model to the lever choice and the latency to choose the lever. I have done 2 different analyses with a much larger data set. So I've got 162 animals in total, almost 50-50 males and females. And the first model analysis I've done is to fit the model to all of the trials within a session. And that's essentially asking a very similar question to I was in the preliminary analysis for the gambling task, which is, are there overall differences in decision making, either between workers and slackers or between males and females, in the way decisions are made about hard or easy choices? What I've also been able to do, because this is a much larger data set, is to also fit the parameters split by whether what happens on the trial after the choice is made. So, for example, I will fit a set of parameters to trials where a premature response is made after a choice, to trials where a correct, so the rats accurately detect the stimulus light, or to those trials where they don't detect the stimulus light. And what I was trying to get at here is to ask, so our assumption is that what rats are choosing when they choose the hard or easy choice is related to sort of their later performance in the task. But, that is an assumption that we've made, and we don't have any good way of kind of sort of proving that particularly given that, accuracy measures and other measures are the same between workers and slackers. And so in this case, the upper boundary is a decision for a hard choice, and the lower boundary is a decision for an easy choice. And so again, I'm going to walk you through these, like each graph step by step, again, starting with the drift rate. And in a very similar way to with the gambling task, we find that workers have positive drift rates going up towards the hard choice boundary, which we would expect because that's how we're classifying workers, and slackers have less positive drift rates. We also find a difference in starting point, where workers have a starting point that is actually biased towards the hard choice trial, so their preferred option, and slackers have neutral starting points. We find that workers have wider decision boundaries than slackers, which kind of also makes sense in that, you know, this kind of suggests that workers are exhibiting a greater response caution, want to be more like sure of their response. And we don't find any differences in non-decision time overall when we look just between workers and slackers. In males and females, we get a sort of interesting difference between, I haven't shown the data for workers and slackers because there's no specific male and female worker and slacker differences. We just get overall differences between males and females. And so what we actually see is that in males have very slightly more positive drift rates than females overall, but to sort of counteract that, they have females have slightly more positive decision starting points. And so again, this is essentially two different ways of, you know, the behavior looking the same, but two different decision making, like the processes that are underlying this are slightly different between the sexes. And so what I then went to have shown with the by trial performance is that decision making is indeed linked to later task performance. And there are a couple of interesting differences between workers and slackers. So again, this is sort of as would be expected for correct trials. There is a sort of more neutral drift rate, and that's basically being driven by the fact that rats that make more easy choices get more trials correct because the easy choices are, you know, they're easier to do and they are more accurate on those. Similarly, for incorrect trials have much steeper drift rates, and this is again because incorrect trials tend to be after the hard choices, which are upwards towards the hard boundary, and premature choices are somewhere in the middle, which makes sense because there isn't actually, I went back and looked, and rats pretty much equally make premature responses after a hard choice or an easy choice. So it makes sense that this would be halfway between the two. But what is interesting is that there is a difference for both the correct and premature drift rates between workers and slackers in the same direction as we'd seen in the previous overall analysis, but no difference in the incorrect trials, suggesting that either there's a ceiling effect here or that it doesn't matter essentially if you're a worker or a slacker, your drift rate is equally strong on those different trial types. There's no differences in starting point, but again, we see sort of an interesting difference in the boundaries. So with correct trials, we have the lowest boundaries, and incorrect trials, the highest boundaries. What we think that this might speak to is that, oh, and a slight difference, so incorrect, so in slackers, the boundaries are slightly even more wider than in, in workers than in slackers. And what we think this might speak to is potentially pulling out a link to essentially just cognitive resources. So, you know, cognitive resources aren't infinite, like there is a limited amount of cognitive resource that a rat is able to sort of apply to a particular trial. And so if on a trial it you know, the boundaries are further apart, there's had to have been more evidence accumulate. It's taken longer for that evidence accumulate. That's presumably taking more cognitive resources, and therefore on those trials the rat maybe then has less cognitive resource able to apply to the attentional part of the task, and then is more likely to get that second part of the task wrong. Again, that is speculation, but something that we could sort of test based on these model results.
Oh, sorry.
Yeah.
I was just going to ask about the correct versus the correct on each side, workers versus slackers.
Mm-hmm.
So you see that significant difference there, and then also in the premature responding between workers and slackers, it looks like there's a significant effect there as well. So I'm trying to understand, is it like they're doubling down the slackers, like if they win on correct ones, or am I interpreting that wrong in terms of their drift rate?
For the drift rate, yeah. So
what would that like? I'm trying to picture in my mind that the drift rate, them being lower, does that mean that they're more likely to kind of double down on a, I don't know. You tell me. Yes.
Yeah. So I mean, it could be that, but the sort of the caution that we have to have with interpreting this is that, you know, slackers are making more easy choice trials anyway. So, you know, that it's much more likely that it is just because of the fact that slackers are choosing the easy option more often, which is going to have a negative drift rate. And so when you average it overall, you end up with a drift rate that is less positive.
Okay.
Yeah, so I mean, the fact that there's a difference between correct and in workers and slackers between the correct and the premature is what we found on this previous, it's the same here. The fact is the drift rates are lower. The interesting one is that there isn't a difference with the incorrect, because my assumption would have been that it would have just been the same across all trial types, because of the fact that, slackers don't show any differences in accuracy compared to workers on either hard or easy choices. So the fact that there is a specific difference in the incorrect trials that is the kind of, the unusual result there, as it were.
Awesome, thank you.
So yes, so I mean, this graph is not the behavioral data from the rats that I have, but generally, slackers are still choosing the hard option more than 50% of the time. So a majority of the time, just less than workers. But I'm going to come to your point in a second on the next slide. That there will be something that will speak to what you kind of discussed. So yes. Oh, so sorry, yeah, for people on Zoom, the question was like, how much do slackers actually choose the hard choice compared to workers? Which I will get interesting results in a second. And so so again, coming back to Ryan's question about the premature sort of non-decision time, this is a completely different task, and we're pulling out the same effect here as well, where for premature, specifically on trials that are followed by a premature response, we're getting longer non-decision times, although this is actually purely being driven by slackers. And so this was actually the first result that I found, which is why we think that maybe this longer non-decision times are representing a kind of less focused, more distractible kind of phenotype, because we see it in slacker rats that aren't as given that they have reduced boundaries, they more often pick the easy choice. We could hypothesize that they are less focused and more distractible in the task potentially. And so
could it be that they're more flexible, or do you think distractible?
I mean, yeah, like that could be another interpretation as well. I will provide some more evidence in a sec though for why we think it's specifically more distractible or like worse performance. So one of the things that we sort of was something that I was particularly interested in is, as I said, the lab initially decided to classify workers and slackers just based on the mean group performance. Overall, on average, animals perform at 70%, and so we use this as a median split. But is this actually a true classification? Like, are we missing? Are there just workers and slackers? Are there potentially more groups within this? Are we oversimplifying? Are we missing something? And so what I've done with this behavioral data set is to use k-means clustering to essentially, let the data tell me, like, is the clusters that we're the two clusters that we have kind of grouped them into, is this real or not? And so for this clustering analysis, I've essentially put in both behavioral measures and model parameters that are relevant to the sort of performance that we're looking at. So the percentage hard choice, which we use to classify workers and slackers to begin with, but also how many choice omissions animals make, how many premature responses they make, the percentage accuracy following both hard and easy choices, as well as the model parameters. So the values for drift rate, starting point, boundary separation, and non-decision time. And essentially, this was sort of motivated by me looking at the data and being like, I think there might be more populations in here based on the diffusion model parameters than we're actually, have been pulling out before. So I wanted to kind of use a method to sort of look at this. And it turns out there are. So the most, I've tried a few different kind of clustering kind of sizes and groups, and essentially found that the most parsimonious explanation is using with a cluster of 4. And so these 4 clusters I have termed good workers, poor workers, good workers, and good slackers and poor slackers. And so just focusing to begin with on the hard choices, these 2 worker clusters have more hard choices than the 2 slacker clusters, which is, essentially what we would hope, or, is why I've still called them workers and slackers. But you can see this really big difference between slackers, you know, the proportion of animals where they are slackers that actually pick the hard reward quite often, so, you know, more than 50% of the time, versus this group of slackers that pick the hard choice like less than 50% of the time, so quite infrequently. So now jumping over to the two accuracy graphs, this is the bit that I found kind of really interesting, was that in terms of accuracy after a hard choice trial, the good workers perform the best out of all the animals, but the poor workers actually perform the worst. So even worse than both of the slackers group. So they, even though they're choosing the hard choice more often, they're actually doing really badly at it. This result also pulls out for workers with on the low reward. So, you know, the good workers are, again, the most accurate on easy choices, but the poor workers are the least accurate out of all the animals. And in this case, for the easy choices, the two slacker groups are actually the same. So, you know, and which is also quite interesting that these slacker animals are picking the easy choice way more often, but don't actually end up being any better than the animals that are picking it like less frequently. But we do see a difference in the accuracy for a high reward between these two groups. I'm almost kind of surprised that this accuracy is not worse, given that they pick the hard choice like pretty infrequently. But yeah, I mean, they are definitely worse than their kind of good slacker counterparts.
I just want to mention anecdotally, because I have been working with Kath and with Beck on some kind of translation of some of this rodent work into human tasks, kind of not exactly the same task, but kind of a similar task involving working memory. And we've just been looking at some preliminary results of like workers and slackers and humans essentially. And one thing that definitely stood out to me is we definitely had a good proportion of, well, slackers who were trying and going for the high effort trials, but consistently failing. But at that point, we were just making the original splits like you were, kind of the original mean splits. So I'm really curious to see if these groupings that you're talking about here actually may be also visible in humans as well. We might actually be seeing some of those similar patterns in humans on a relatively, I guess, comparable human task. Yeah, that's really cool.
That for sure would be really cool to know. That was kind of what I was going to go on to say after this, is like, now I'm really interested to know that, for example, like, is it the poor worker group that are particularly susceptible to, you know, sort of others, like would stress make that exacerbate this and it's specifically these animals that would be more affected? Yeah, I don't know, but it would be really cool to know if there was a similar thing in humans.
Yeah, and we are looking at stress as well in humans. So yeah, that's so cool. Yeah.
And so the sort of other interesting thing is that, I mean, I guess it's not interesting, it kind of makes sense in a way, but that, you know, there are differences in the decision-making processes that are driving the performance between these groups. So again, as with, you know, all of the drift rate analyses, this step pattern matches what we see up here with hard choices, which, you know, should do, because that is the definition we're kind of using to classify these groups into workers and slackers. But what is more interesting is that this sort of poor workers are using a quite different strategy to obviously overcome or, you know, perform the task by choosing these kind of hard choices, and maybe, you know, the way they're sort of getting around this effort cost is quite different to the good workers. So the good workers have pretty much neutral starting points, but these like high drift, you know, strong, steep drift rates. Whereas the poor workers are almost kind of my interpretation is almost like they're cheating. Like they've got this bias, like predetermined bias towards the high reward boundary, which means they don't actually need to, you know, the evidence accumulation doesn't need to be as steep because they're starting closer off to that boundary in the first place. But maybe this is the reason that they are actually then so much poorer at it, is because they're not, you know, even though it looks to us like they're putting in the same amount of cognitive effort in choosing the hard choices, actually, you know, the cognitive resources that they're using to make these decisions are potentially quite a lot less, because they're needing to accumulate less information to get to that boundary. We see essentially the boundary, you know, result isn't super interesting. It matches what we saw before, that the two worker groups have wider boundaries than the two slacker groups. But what is interesting, which is coming back to this whole, you know, is the non-decision time. Could it be flexibility? Is it just that they are, you know, is it distractibility? Is that it's only in the poor slacker groups that have a significantly higher non-decision time, suggesting that it is, you know, these animals that are sort of not putting the effort in on the task are also potentially the ones that are will have these longer non-decision times, so maybe are less focused and more destructible, more influenced by extraneous factors than the other groups. And so yeah, what I kind of really want to sort of look at, the questions that would be really interesting with this is, does this kind of, are any of these groups sort of the ones that are sort of susceptible? Is it equivalent in humans? Like, what is the sort of kind of crossover here?
Can I ask a question about the poor slackers?
Mm-hmm.
Do you have like any kind of control or anything that you could do to see in terms of how they value the reward magnitude between the small and the large reward? I'm just wondering, could that be something driving some of your effects?
Yeah, I mean, it potentially could, for sure. Like, it could just be that they don't either, the 2 pellet reward to them isn't as, they don't value it as much. It isn't twice as valuable to them as the 1 pellet reward. This is all data from rats that are long gone now, so I can't tell from this specific data set.
I guess I'm just thinking about
forwards, like we could.
I'm just thinking about, like, if I'm at the casino and I had a couple of drinks, do I bet bigger? Do I bet smaller? Do I go safe? And then I kind of, sometimes it's like it doesn't even matter. It's like it's almost like you're treating it the same, and then so you'll maybe go more risky, or you're just like kind of. So I guess this is anecdotal though. Like I said, I don't know much about this research, but it's really awesome stuff. But I'm just wondering if there's anything with that at some point that maybe the magnitude of the reward is valued differently.
Yeah, so I mean, it definitely is something we could test in future cohorts. It's quite easy to just do a reward magnitude. You can, without any of this extra effort involved, you could give rats choices between 2 pellets and 1 pellets and see how often, how quickly they press levers for them. But the thing that the one really cool data set that we have that maybe could speak to this a little bit is we have a cohort of animals that were trained on both the gambling task and the cognitive effort task. And so just based on the sort of pure behavioral measures, so Brett trained these animals at Grad Student Lab, and he hasn't found any like super strong, there are some kind of correlations there between a worker's more risky or more optimal, but there wasn't anything that kind of like super kind of jumped out. But what I am now going to do with that data set is to kind of use this sort of clusters that I found, and maybe it's actually that just using workers and slackers and optimal and risky, we're not getting fine-grained enough in a single cohort of animals to have the power to see these associations, but perhaps it is that actually poor workers are specifically the very risky rats, but because they're only making up kind of roughly 50% of the whole worker population, you miss it when you're only looking within one cohort. So that is sort of something I want to go on to do that could then speak to that, because if it is, for example, the poor workers or the poor slackers that are always picking P2 versus P3, or the different reward magnitudes on the RGT, that might suggest that they have different reward valuation mechanisms. The other thing based on this that I now want to sort of do, which is kind of what I was going to go on to say, like kind of the summary, is essentially to do the same kind of clustering analysis in the RGT. So we have lots and lots and lots and lots of data from the RGT, like tons and tons of cohorts. And so, is it the case that actually we are classifying optimal rats as being those that make, more optimal choices than risky choices? So essentially a 50-50 split, but actually is there multiple groups? Like, are there sort of, super optimal rats that are actually performing, making decisions quite differently to super risky rats? Are there rats that are pretty much neutral in the middle? I mean, we would kind of expect that these might be doing something different to the really optimal versus the really risky, but maybe there are even groups within the sort of more neutral rats in terms of the processes that are driving their decision making. So yeah, just to sort of summarize, hopefully I've shown that we can use modeling to provide some more insight into the sort of cognitive processes that are driving these differences in decision making across tasks between groups, and really interestingly between sexes. I think this is something that I want to kind of dig down deeper into, because it is definitely the case for both of these tasks that the behavior looks the same based on just the standard analysis of behavioral measures at baseline, but there is actually these kind of differences in decision-making processes that the models suggest are there. And so yeah, I'm kind of going to skip the sort of rest of it, but you know, one thing that I am really a big proponent for is that, you know, using modeling can provide us with much more insight than these traditional analysis, because we're finding things that, you know, we just can't see with the behavioral data. And so, you know, some of the sort of things that I want to go on to, I've kind of already said, but a couple that I specifically wanted to point to as well is, well, yeah, extending the RGT modeling, you know, putting more animals in, doing this clustering analysis. But one thing that I'm really kind of interested to see if I can use modeling approach to provide some insight into is this really quite interesting phenomenon we get where we have, so this is essentially graphing performance across session, where on every choice that a rat makes, they get a +1 if they make an optimal choice and a -1 if they get a risky choice. And you can pretty much see with these optimal rats, like a lot of them just keep getting better, like keep learning, keep making more optimal choices. But what's really, really interesting, I think, is that you have some rats that start off getting more optimal, and it's like they're figuring it out, they know what they're doing, and then for whatever reason there's this like turning point where they kind of switch and then they just start getting more and more risky. And even like some animals, like this kind of gray animal that is going risky, and then it kind of takes a little bit of a turn. It's like, oh, I'm being more optimal, I'm getting it, and then no. And so, you know, something that I'm really interested in sort of seeing is, you know, what is it around these change points that is changing the decision process? Like, is it that, you know, is it the case that there are different strategies being used, which is kind of what I'm kind of hoping there is. Then, you know, the rats are using one strategy and then actually suddenly they're switching to this kind of much more risky decision-like strategy. And then on the cognitive effort task, which is kind of coming back to what my very first motivation from the start is, that we actually have human data from the exact same task with giving humans amphetamine as we do, and we've got these manipulations in rodents as well. And so I am waiting for the human data, but what I'm hoping to do is pretty much a replication of what I did in my PhD, where we've done the just translational task, same manipulation, in this case the exact same manipulation, we've given them both amphetamine, and can we use the model to sort of show, are we measuring the same thing? You know, we're kind of assuming that this cognitively effortful decision-making task in rats is measuring something relevant, like is it measuring the same thing? But also in both the rat and the human data, we see these quite interesting sex differences between the effect that amphetamine is having on males and females. Is that being driven by a similar difference in cognitive processes as I'm seeing at baseline, or is it being just even driven by the same process in you know, male rats as it is in male humans, or is it something different? So yeah, with that, I just want to thank all of them in Stanley Lab, especially all of those students that have collected the data that I'm using. So I haven't had to stand, you know, put all these rats in boxes, which is great. And the funders. Yeah, if anyone's got any questions, I'm happy to answer.